Imagine your gut, the very core of your well-being, slowly turning against you as you age, becoming a breeding ground for cancer. Sounds terrifying, right? This is the reality researchers are uncovering, and it all boils down to something called epigenetic drift. Scientists at the Leibniz Institute on Aging, Fritz-Lippmann Institute, FLI, in Germany, along with collaborators in Italy at the Molecular Biotechnology Center, MBC, and the University of Turin, have pinpointed a key aging mechanism within our intestines. Their groundbreaking research reveals that as we get older, a specific type of epigenetic aging, known as ACCA drift, aging and colon cancer associated, accumulates in the stem cells residing in our gut. But here's where it gets controversial. This isn't just random wear and tear, it's a targeted shutdown of vital genes through a process called hypermethylation, essentially, adding too many chemical off switches to our DNA. Now, you might be wondering, what exactly is epigenetic aging? Think of your DNA as a complex instruction manual for your cells. Epigenetic markers are like sticky notes attached to those instructions, dictating which genes are read and which are ignored. These markers change throughout our lives, influenced by our environment, diet, and even our stress levels. In the gut, this ACCA drift isn't uniform. It spreads like a creeping vine across the intestinal crypts, tiny, test tube-shaped structures lining the gut wall. This drift is fueled by a nasty cocktail of age-related inflammation, a decline in crucial WNT signaling, and a disruption in iron metabolism. And this is the part most people miss. The researchers believe this epigenetic shift is a major reason why the risk of colorectal cancer skyrockets as we age. The human gut is a master of self-renewal, constantly replacing its lining with fresh cells every few days. This remarkable feat is powered by specialized stem cells. However, with each passing year, these stem cells accumulate epigenetic changes. The research, published in Nature Aging, demonstrates that these changes aren't haphazard, they follow a distinct pattern, the ACCA drift. As Professor Francesco Neri, formerly of the Leibniz Institute on Aging, explains, we observe an epigenetic pattern that becomes increasingly apparent with age. Critically, the genes most vulnerable to this drift are those responsible for maintaining balance in healthy intestinal tissue. This includes genes that govern the renewal of the intestinal epithelium via the WNT signaling pathway, a critical communication network that keeps stem cells active and functioning properly. The changes observed in the aging gut also appear in almost all colon cancer samples examined. This strongly suggests that the aging of stem cells creates a fertile ground for cancer development. This raises a huge question: could targeting these epigenetic changes delay or even prevent colorectal cancer? A particularly striking finding is the uneven distribution of this epigenetic drift. Each intestinal crypt originates from a single stem cell. As Dr. Anna Krepalova elucidates, over time, more and more areas with an older epigenetic profile develop in the tissue. Through the natural process of crypt division, these regions continuously enlarge and can continue to grow over many years. This explains why the intestines of older individuals exhibit a patchwork of crypts, some youthful and vibrant, others aged and degenerated. This unevenness creates zones that are particularly prone to producing damaged cells, which, in turn, fuels cancer growth. But what's the root cause of this ACCA drift? The researchers discovered that older intestinal cells become less efficient at absorbing iron while simultaneously releasing more of it. This creates a shortage of available iron, too, within the cell nucleus. Why is this important? Iron, too, acts as a crucial cofactor for TET, 1011 translocation enzymes. These enzymes are the guardians against excessive DNA methylation, preventing the off switches from being stuck in the on position. When iron is scarce, the TET enzymes can't function properly, leading to a buildup of unwanted DNA methylations. As Dr. Krepalova puts it, when there's not enough iron in the cells, faulty markings remain on the DNA. And the cells lose their ability to remove these markings. This sets off a chain reaction. Decreased TET activity leads to more DNA methylation, silencing vital genes and accelerating epigenetic drift. Furthermore, the study revealed that age-related, low-grade inflammation in the gut exacerbates this process. Inflammatory signals disrupt iron distribution and strain cellular metabolism. Simultaneously, the WNT signaling pathway weakens, further compromising stem cell function. This trifecta of iron deficiency, inflammation, and WNT signaling decline acts as a potent accelerator of epigenetic drift, causing the aging process in the intestine to accelerate faster than previously imagined. Here's the good news: despite the complexity of this mechanism, the study offers a glimmer of hope. The researchers successfully slowed down and even partially reversed epigenetic drift in organoid cultures, miniature intestinal models grown from intestinal stem cells. They achieved this by restoring iron import or by specifically activating the WNT signaling pathway. These interventions boosted TET enzyme activity, enabling the cells to remove the excess methylations. This means that epigenetic aging does not have to be a fixed, final state, emphasizes Dr. Krepalova. For the first time, we are seeing that it is possible to tweak the parameters of aging that lie deep within the molecular core of the cell. This is a game changer. It suggests that we may be able to develop therapies that target these epigenetic changes, potentially preventing or delaying age-related diseases like colorectal cancer. What do you think? Could lifestyle changes, like diet and exercise, play a role in mitigating epigenetic drift in the gut? And if we can reverse epigenetic aging in organoids, how far away are we from doing the same in humans? Share your thoughts and opinions in the comments below.